Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Oded Rechavi. He's at the University of Tel Aviv. He has a lab called the Rechavi Lab. We're going to be talking about epigenetic inheritance and his research. So, Oded, thank you for coming.
My pleasure. Thank you for inviting me.
Yeah, if you don't mind, tell me about your research. What does it involve?
We study many different things. The most relevant thing to this particular podcast is we're using worms that are called C. elegans, which is a very important model for biology, to study inheritance of traits between generations. So inheritance of parental responses or even memories between different generations.
When you say inheritance of memories, is that learned behavior, or what's an example of an inherited memory?
Yeah, so memory, normally when we think of memories, we think about the memories that are encoded by the brain. But there could be other types of memories also. Any lesson that we learn from the past and we change our behavior as a result constitutes as a memory for me. I mean, of course, there are different definitions. But we, in general, are interested in how parental responses to the environment affect not only their responses in the future, but also their progeny's responses. And in theory, I mean, according to regular biology and what we know about DNA-based genetics, that shouldn't happen. But in worms, we now know of a different alternative inheritance mechanism that does allow some responses to be inherited to the next generations, including responses that were triggered by the nervous system of the previous generation, by brain activity.
So what kind of activities did you set up for the worms to do to see if they were inherited? And, when knowledge or memories were inherited, what did that allow the inherited worms to do?
Well, first of all, the first thing that we've shown, and this is something that I started doing about 10 years ago, when I went to do my postdoc with Oliver Hobart in Colombia, was to see whether memory of viral infections can transmit to the next generations. So a sort of immunological memory, although in the worm, it doesn't involve dedicated immune cells like it does in humans. And back then, what we've shown is that when you infect worms with viruses, when you challenge them with different viruses, and we use artificial ones, and later on, people use also more natural viruses. The worm produces an immune response that transmits also to the next generation and persists for multiple generations. And this was very interesting, but what was more interesting is that this was an excellent system to study how such a weird thing could happen. And we found that it happens via the inheritance, not of changes in the DNA sequence, but through changes in molecules that are called small RNAs, which is one type of RNA molecule that regulates gene activity. And after that, we continue to show that also the worm transmits to the next generation responses to starvation, to very strong starvation that they encounter during development. And later, other people have shown that also temperature stress and stress that's caused by different bacterial pathogens can transmit to the next generation. And using all these different assays, we really learned a lot about inheritance of small RNAs, which really follows different rules from the DNA-based inheritance, which is something that the scientific community know for many, many years, since the monk Mendel studied it in his garden about 150 years ago.
So where are small RNAs kept inside the worms? Do they have a repository where they stay, in every cell, like the nucleus? or are they free moving?
So small RNAs, the type of small RNAs that are inherited between generations in C. elegans, in these nematodes, they are acting in the nucleus, and they function by regulating gene activity, by preventing genes from being expressed. However, they have a life also outside of the nucleus, because they need to get outside of the nucleus to get amplified. There's an amplification system for these small RNAs, and this takes place by enzymes, dedicated enzymes that are called RNA-dependent RNA polymerases that use RNA as template to self-amplify. And we've shown in this paper about the virus that these enzymes are needed for transgenerational responses to transmit between generations. And so they function. They are active. They affect gene expression by acting in the nucleus, but they also go out of the nucleus to the cytoplasm, or to specific granules that reside outside of the nucleus. And small RNAs are made in the worm in different tissues. The one to get transmitted to the next generation, they have to reach the germline, because every animal starts its life from the fusion of the sperm and the egg, from the germ cell. So to get inherited, the small RNAs from whatever tissue needs to get to the germline. And what we recently showed is, and this is very related also to the memory question, is that small RNAs that are made in the nervous system can cause transgenerational effects. So we've shown that if you change the population of small RNAs just in the brain of the worm, in its nervous system, this is enough to generate a response that transmits across multiple generations, regulates gene activity in the grandchildren and grandchildren, and also via this control over gene activity also controls the descendants' behavior, their ability to find food, particularly.
Does the effect get less and less over generations if the stimulus is not there?
This is something that we do know happens when you trigger an artificial response. So you can feed the worm, and this was something that was shown 20 years ago by Craig Mello and Andrew Fire, and in the study that they did to identify these mechanisms of regulatory RNAs that eventually that awarded them with the Nobel Prize. Back then, 20 years ago, they've shown that if you feed worms with RNA artificially, this also affects the progeny, the immediate progeny. Later, it was shown to also transmit across multiple generations. And we know that the response typically peters out after a while. And normally, in most of the worms in the population, it stops after 3 to 5 generations. And originally, because there wasn't any better idea, people thought that maybe the RNAs just dilute. The RNAs that the parents make that just dilute until there's nothing left after 3 to 5 generations. But we thought that this is impossible because every worm produces 250 babies. So so the dilution factor across generations just it becomes exponential and totally impossible, because after 4 generations, 250 times 250 times 250 times 250 equals to something like 4 billion, which is completely homeopathic and would never work. Instead, we found that there's a mechanism that limits the duration of the response, and we published a paper about it, about how it works. It's a sort of a repressive feedback loop. And this mechanism limits the response typically to 3 to 5 generations, but we know how to hack the mechanism, to hack this timer, and get responses to last a lot longer, even hundreds of. And also the response stops after a few generations at the population level. But very recently we published a preprint that shows that there's big difference between different individuals in the population. Some of them have very long-lasting responses, and some very short-lasting.
So have you been able to figure out the inception of, you expose the worms to a stimulus and then they start to change, it manifests in their small RNA at some point, but have you been able to watch the cascade or the process and figure out how it starts and how it finishes?
It depends on what it means to actually watch it. So we found that exposing the worms to different environmental stresses leads to changes in small RNAs that also transmit to the next generations and affect the next generations. Exactly how the environment changes the endogenous small RNAs that the worms makes is still a big question, and we don't know exactly. When it comes to starvation, we found that exposing the worms to starvation lead to the production of small RNAs that regulate nutrition-related genes. And this could be because the transcription of these nutrition-related genes somehow leads to their silencing or to their creation of small RNAs that regulate the same genes. It's possible, but there are still many unknowns about how small RNAs are made in response to particular stimuli and particular challenges.
So what's your near-term goal? What are you looking at right now to try to figure out?
So there are many open questions. We want to know more about the heritable small RNAs, how they regulate behavior in the next generations. In our last paper, we identified one gene target that's regulated by transgenerationally inherited small RNAs that start in the brain, which affects behavior, but we don't know how it continues from there. How does it affect behavior? So this is something that I want to study. A big question that we're studying, which is more long-term, is can these processes affect evolution? This is an alternative inheritance mechanism, and it's not clear at all whether it can change evolution, the process of evolution or not. Normally, when we think about evolution, we're thinking about long-lasting about responses that take many generations. And here, most of the responses stop after a while. Can this process nevertheless change the process of evolution or speciation, or can it become hardwired in the genome, in the DNA-based genome? These are the questions that I'm most interested in. But there are so many open questions, really, that will be busy for a long time.
If you consider evolution instead as like a continual adaptation, it seems to make sense that these organisms would be exposed to stimuli, they would change themselves and adapt. At least on the small RNA level. Do you see any changes at the DNA level?
Not yet, but we're very interested in looking for that now. So this is something that we're actively looking at to see whether changes in small RNAs can somehow change the DNA.
Well, again, how would a small RNA first come into existence? It's created in various places in the cell, but again, have you been able to look into the early stages of the process somehow,
right,
and see the first creation of these?
Right. So there are different types of small RNAs. Some small RNAs, like microRNAs, those who know a little bit about the biology of non-coding RNAs, they are acting like genes, regulated like other genes. Means they have promoters that are activated by transcription factors that can be that can be expressed in response to different stimuli or developmental stages and so on. So just like regular genes. Other small RNAs, like the one that get inherited between generations, like endogenous siRNAs, for example, they are made, so they tile the entire length of their target genes. And they could be made in response to expression of their targets, or maybe in response to expressions of somewhat aberrant targets that look foreign to the worms. It's not exactly clear, but there are all kinds of feedback loops that regulate gene activity, and small RNAs seems to be just one of these players that function in gene regulation, and they can be regulated themselves on many different levels.
So are you hoping to find out if there's changes in the underlying DNA? Is that your next step, or what?
Yes, this
would
is
be a big breakthrough for you?
So this is definitely one of the goals. We want to know what are the limits to RNA inherited. So what type of responses can be inherited? What's shared between the different responses that are inherited? And really do also to check the link to evolution, to do lab evolution experiments, to watch evolution as it happens, and to see whether small RNAs play a role in that.
Are there viruses that affect worms or bacteria that affect them or live, as part of their microbiome?
Yeah, so surprisingly, until 2010, there wasn't any known virus that infects C. elegans. In 2010, a natural virus that infects C. elegans, that's called Orsay, was discovered in France. And it was discovered in the wild isolates of C. elegans. But the worm that they found was defective in small RNA production. This is why it was susceptible for the virus. The worms that we work in the lab in are more resistant to the viruses, and it's controversial whether there's any antiviral inheritance when it comes to this particular virus, the Orsay virus. Other viruses that are more artificial was shown to trigger transgenerational silencing by small RNAs. Aside from that, from this Orsay virus, there's no known virus that infects C. elegans, but there probably are some. There are viruses that are being discovered that infect other types of close relatives of C. elegans or other types of nematodes. So I'm sure there are also viruses, additional viruses that infect C. elegans, perhaps even viruses that evade the antiviral immunity, that's the small RNA-based antiviral immunity. There are certainly pathogens, other pathogens that infect C. elegans and successfully do so. And so there are different pathogenic bacteria. And also recently there were a few papers in the last couple of years that showed that exposing to worms, to bacteria, can generate transgenerational responses in the form of small RNAs, heritable small RNAs, and also changes to the chromatin.
Do C. elegans appear to have their own microbiome? A commensal microbial attachment that stays with them?
Yeah, so people study that, and I think the answer is yes. I think the gut content reflects their environment maybe more than it does in humans, but there still seems to be commensal bacteria that are present there. And we can, in our experiments, we can control for responses that are affected by the microbiome simply by bleaching the worms between generations, and this kills the adults, the mothers, and also their bacteria, and just the eggs survive because of their hard shell, which is protected from the bleach. So this is a way to eliminate any artifacts that might be caused by the microbiome. But it's possible that there's also transgenerational transmission of bacteria that also lead to these interesting non-Mendelian, non-DNA based responses.
Yeah, it's very interesting. So what's the best way for people to learn more and to read papers you put out and maybe get in contact?
So there are many. There are many papers. There are also recent review papers that we and others wrote. That could be nice entry to this field. I think we wrote, me and my student Itamar Lev, a review 2 years ago in Current Biology, which I think nicely summarizes a lot. Although there are also new papers. So you can go on my website and see the papers that we published and learn from that. I recently gave a TEDx talk in Vienna that's online, and I think this is a very simple way to just get the gist of it. And there are very interesting discussions on transgenerational inheritance. What's established, what's controversial, also going on Twitter. So I think there's lots of action on Twitter and many very active discussions that are worth looking into.
Yeah, just last question. Do you think all this goes on in humans to a greater extent, less extent, not at all? What's your thoughts?
I think we just have no idea. We can do all kinds of guessing and speculate, but this is hard to study, and it's just the beginning. I mean, in C. elegans, the power of studying this in these worms is just the generation time is just 3 days. So we can follow many generations very easily and quickly. And every worm produces 250 babies, so you have the statistics to do it. And also worms, they are isogenic, they are genetically identical almost. So you can really control for nature versus nurture and DNA versus non-DNA changes. In humans, it's so difficult. It's so difficult to do that, we need to even think of how to do it. So we just have no idea. Some people speculate that it might not happen in humans because all kinds of reasons. For example, the chances that the progeny of worms will experience the same environments that their ancestors experienced are relatively high because they don't go very far and because not so much time passes. With humans, your kids could be traveling to a different country, and also it takes 20 generations, and 20 generations at least separate between generations, so many things could change. But that's not a good enough reason, and there are things that I can imagine that would transmit nevertheless. Some of the machinery that we know that enables small RNA inheritance in C. elegans doesn't exist in humans, like these RNA-dependent RNA polymerase that we've shown in the viral paper to be important for transgenerational inheritance. But that doesn't exclude the possibility that humans do inherit responses, because there are many other feedback loops that could perpetuate heritable silencing aside from RDRP. Changes in methylation, changes in different chromatin marks, many different things. So I think it's definitely possible. We just have no idea because it's so hard to study.
Well, very good. Oded, thank you for coming. I appreciate it. It's a really fascinating area. We'll see what comes. So thank you.
Thank you very much. My pleasure. Thank you.
Forget frequently asked questions, common sense, common knowledge, or Google. How about advice from a real genius. 95% of people in any profession are good enough to be qualified and licensed. 5% go above and beyond and become very good at what they do. But only 0.1% are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in fields such as sleep science, cancer, stem cells, ketogenic diets, and more. Get ready. Here come the geniuses. This is the Finding Genius Podcast with Richard Jacobs.